Leading a startup team? Whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. Werner's and Hutchinson-Gilford Progeria syndromes are rare genetic diseases that cause children to appear to suffer from advanced aging at young ages. The syndromes are of particular interest to researchers because of the potential they may have to help understand the biology of normal aging and lead to ways to counter aging-related diseases. We spoke to Brian Kennedy, President and CEO of the Buck Institute for Research on Aging, about his Progeria research, the potential to repurpose existing drugs to combat these syndromes, and what insights into normal aging are being gained from the study of progeria. Brian, thanks for joining us.
Oh, anytime. Happy to be here.
We're going to talk about aging, a rare childhood disease known as Werner and Hutchinson-Gilford Progeria syndrome, and what one may tell us about the other. Let's start with progeria. What is it? How rare is it? How does the disease manifest itself? And what is the prognosis for someone who has it?
It's an extremely rare disease. It affects about 300 or 400 kids worldwide. Typically, they get diagnosed in their first or second year of life because they have a failure to thrive. And this gets progressively worse. And the interesting thing from an aging perspective is they seem to manifest with certain properties of aging, and they don't have others. And so they get cardiovascular disease, they get atherosclerosis, they tend to die in their teens of heart attack and stroke, but they don't have neurologic disease. In fact, they're quite precocious, smart. So people have been interested in this disease for a long time, but not only in an effort to try to help these kids, but also to try to understand how it might relate to normal aging, the rest of it.
Are there any treatments for progeria today?
There is one drug that's approved, and it's a farnesylation inhibitor, farnesyltransferase inhibitor, and I can explain a little bit about that if you're interested. But it has a very modest impact. The kids do a little bit better with it, but it certainly doesn't cure the disease. The average life expectancy is in the teens, around 14.
What is known about the underlying biology of the disease? How well understood is that?
Yeah, it's interesting because the kids have a mutation in this gene called LMNA, which encodes a protein called lamin A. This lamin protein is a structural component of the nucleus of the cell, so it helps organize the nucleus so that transcription and DNA replication and maintenance of DNA can take place in an ordered fashion. When you have this mutation, it actually is a dominant mutation. It causes lamin to do things that it's not supposed to do, and that disrupts several different activities in the nucleus of the cell.
Obviously, you're a researcher focused on aging. How did you come to become interested in a childhood disease? Is it a good model for aging as we know it?
I think that the normal aging field has debated whether these progeria models are good models to study aging. And I'm kind of strange because I was working on aging, and I also had a separate project at the time about 15 years ago looking at these lamin proteins, which at the time had nothing to do with aging. So it was just fortuitous that that mutation was identified by Francis Collins, who's now the NIH director, uh, in lamin A that causes progeria. Because at that point I was like, well, I'm going to look into this because I'm already working on lamins, and I'm curious if there are links between aging and this disease process.
So, do we know what role lamins play in normal aging?
Uh, Not sufficiently. So if I could talk a little bit more about the specific mutation in the progeria, what it does is it causes activation of a cryptic splice site. So I need to dig in a little bit here. So you make a long RNA from the DNA, that's transcription, and then you cut out pieces of the RNA before you use the RNA to make protein. And how you cut out pieces of the RNA influences what protein is made. So in this case, you have a mutation that stops cutting out a certain part of the RNA, and you make a protein that misses essentially 50 amino acids at the end of the protein. Now, the reason I'm telling you all that is that there's some evidence that this splicing alteration actually happens in normal individuals as they get old. So as we get old, we may make a protein that is just like that progeria protein that the kids have. And if that's the case, that could be contributing to aspects of normal aging. But I still think we still have to validate aspects of that. And so I think at this point I remain somewhat agnostic as to whether the lamins are affecting normal aging or not.
You had mentioned earlier a drug that's used to treat progeria today. Is this lonafarnib?
Yeah.
So this drug was originally developed to treat cancer, I believe. Do you think this holds promise, and is it understood what that's doing?
Yeah, well, we know a lot about what it's doing. A lot of proteins, after they get made, they get a lipid group called a farnesyl group put on the C terminus. And that often helps the proteins get targeted to membranes, which have a lot of lipids. And so for a protein like RAS, which is a cancer protein, a signaling protein, it gets farnesylated, and people developed farnesyltransferase inhibitors to block that process with the idea that they could stop proteins like RAS from being hyperactive and be cancer therapies. It turns out that those drugs don't work especially well for cancer, although they are used in some cases. But the lamin A gets farnesylated as well, and that probably targets it to the membrane around the nucleus, the nuclear membrane. And it goes through a similar process. So the idea was that if you could repurpose these farnesyltransferase inhibitors to block farnesylation of lamins, and that might be a therapeutic approach in these progeria kids. And indeed, if you look at mouse models of the disease or the kids, the clinical trials with the kids, it does seem to help, although the effects are very modest.
The thought I take it today is that it may not be great on its own, but it may work as part of a cocktail of drugs.
Well, I think that it's kind of you don't really have a choice the way clinical trials are set up. So when you do a clinical trial, you have to do whatever your new drug or therapy is compared to current best of care. So effectively, that means you're going to do the clinical trial with lonafarnib and another drug. So I would guess that the new therapeutic approaches that are being developed, and I can talk about some of those things, that they're going to be done in combination with lonafarnib. And it's true that you might see synergistic effects by targeting multiple aspects of the pathway. Yeah, you could see. It could be that for this disease, and actually for a lot of diseases, that you're going to need multiple drugs to really have a major impact.
Well, you're conducting studies into two small molecules that have garnered interest as agents that might combat age-related chronic disease, rapamycin and resveratrol. I thought we could walk through each of those and what implications it might have for progeria. What is rapamycin?
So rapamycin is a very specific drug that inhibits this kinase, a protein kinase called TOR, and it's made by a bacteria on Easter Island, so that's why the name Rapa. and mice because it kind of has antibiotic properties toward fungi. But people got interested because Tor is often active in various cancer states, and so they thought rapamycin. This was decades ago. They started studying rapamycin to see if it would be a cancer drug. And in fact, it is approved for some rare forms of cancer, but again, much like the farnesyltransferase inhibitors, it's not as effective as people hoped. And part of the reason for that is that cancer cells can figure out ways to get around this Tor inhibition that you get from rapamycin. So, but it's a very specific drug, and our lab and others were about in the last decade have shown that turning down this Tor kinase extends a normal lifespan, and that works in yeast, it works in worms and flies and mice. And so rapamycin has been tested a lot in mouse models of aging, and you can get a 30% extension of lifespan with rapamycin. And as we might expect for an aging drug, it affects many diseases as well, because aging is the biggest risk factor for most of these chronic diseases. So if we can slow aging, we can delay the onset of these diseases, and that's what rapamycin does. So it's kind of working backwards. In this case, we're saying the concept is that you have a drug that affects normal aging, then it might be useful for these kids with progeria. And the published data, mostly from Francis Collins' lab and collaborators, people postdocs that came from his lab, are showing that you can reactivate autophagy when you treat these progeria cells with rapamycin. So, for reasons that are not clear, if you have this mutation in lamin A, it blocks or reduces autophagy, which turns over damaged proteins in the cell. And we've known for a long time that Tor regulates autophagy. Tor, in fact, inhibits autophagy. So if you block Tor with rapamycin, you can reactivate autophagy. So the idea would be that it might be helpful with the kids, and we've been trying to test that in mouse models of disease of the progeria.
And for a non-scientist, what is autophagy?
Yeah, so this is a process by which damaged molecules get recycled in the cell. And so when you have a misfolded protein, it gets sent to this organelle called the lysosome, where it gets recycled into amino acids. And it can be even a damaged organelle, like a mitochondria, that gets targeted to the lysosome for recycling. So autophagy is that recycling process. It's really important for cells to function because it allows you to get rid of the dysfunctional proteins that are accumulating, and some of those can be sources of oxidative damage, and so it's kind of the trash collection system of the cell.
So one of the thoughts about the process of aging is that there's a breakdown in the ability of cells to clear themselves of cellular waste.
Yeah, and it may be that for normal aging, rapamycin works, at least in part, through activating this autophagy system.
So in the case of progeria, could the same be said? Is that part of the thought of what's happening, that cells aren't clearing themselves of metabolic waste?
Well, that's what that data in cell culture would suggest, but I think that we really don't know that for sure. To what extent altered autophagy is affecting progeria in the kids and in the mouse models, and that's something that needs to be addressed in more detail.
Another compound you're looking at is resveratrol, the magic substance in red wine that people have long looked at for its properties of potentially countering aging.
Yeah, let me start with that statement about wine. I think the first magic substance in red wine is ethanol, because almost all of the studies that have been done looking at alcohol consumption and mortality rate in humans suggest that moderate alcohol consumption is beneficial for aging. Now, excessive alcohol consumption is a bad idea, but we're talking 1 to 2 drinks a day. And then there's some evidence that suggests that red wine is better than other forms of ethanol. And that's due to, or possibly due to, these polyphenolic compounds that are in the skin of the grape. and resveratrol is one of those compounds, but there's actually a lot of them. In any case, resveratrol has been tested because it activates this protein called SIRT1, and SIRT1 is the more activity that you have, at least if you're an animal model, the better you age. So again, resveratrol is sort of a potential longevity compound, and so that's why it was really considered in the Progeria. So we were collaborating with a lab in Hong Kong that was studying a mouse model of Progeria, and they showed that a couple things. One is that mesenchymal stem cells become dysfunctional in their mouse model of Progeria, and that when you treat with resveratrol, you restore the function of these stem cells and you extend the lifespan of that mouse model. And so we've been testing it in a different model of Progeria that is an exact copy of what happens to the kids.
Not to be a wise guy, have we tried giving these kids ethanol?
No, but I think that the funny thing about ethanol, we're starting to get into this, but studies not on the Progeria but in normal aging, because we really don't know why ethanol is beneficial. There's been a very limited number of studies in animal models. That's one of those rare cases where we have good data in humans, but not much data in animals. So we need to go back and figure out mechanistically what's going on.
Well, what do we know about resveratrol in Progeria patients?
Well, nothing in patients. To my knowledge, there is a small clinical trial that was started in China, but I think there are only two kids that have been enrolled, and so there's not definitive data yet. And of course, rapamycin is being tested clinically by Leslie Gordon and colleagues on the East Coast, and so that'll be interesting. That's a much bigger study, and I don't think there are any results for this yet.
Well, mechanistically, is there reason to believe resveratrol might have benefit for Progeria patients?
Well, the idea was that in this study in Hong Kong that the lamins that I talked about are controlling SIRT1 function, which might be a target of resveratrol. So when you had the Progeria mutation, the SIRT1 enzyme was dysfunctional, and you can restore that by adding resveratrol. So yeah, there is a mechanistic argument for why it might work, and our data in the other mouse model is not as positive. So we're trying to figure out if we need to use other strategies to activate SIRT1 or do different dosing with resveratrol right now to get a positive response.
What is the status of your research into these compounds, and what's the path forward in finding a way to slow or reverse the progression of Progeria?
Well, so the data in our hands with the resveratrol is not positive right now, so we're still trying to think about how to use other ways to activate that pathway. For rapamycin, we have kind of confusing results at the moment. So we started off with a relatively low dose of rapamycin. We find that most mice are not affected by it. There are a few mice that seem to do better, and then there were a couple mice that seem to do worse. So, we're trying to figure out why the mice respond differentially and how to get all the mice responding better. I think that these things are always challenging because mouse models they're not as expensive as clinical trials, but they're still expensive. And so, what you'd like to do is to try 5 different doses of the drug, but it's just hard to find the money to do those kinds of studies. So we're still trying to optimize, put it that way.
Progeria is an ultra-rare disease. People sometimes question the investment and effort into finding therapies for such conditions, but aging, with any luck, is something everyone stands to confront. This is an example, I suspect, of how research into a rare disease can give us insight into more common ailments. Have you gleaned any insights from this work to help us come to a better understanding of aging and how to combat its negative effects, or do you expect it to yield that?
I do, and I think we've already learned that lamins are now a candidate to regulate normal aging, and there's some evidence that's happening. And there certainly need to be more studies on that. But I think that in this case, progeria is really starting to inform us about aging. Now, progeria is what's called a segmental, Hutchinson-Gilford disease is what's called a segmental progeria. And like I alluded to earlier, what that means is that some aspects of aging seem to be accelerated, but not all of them. And so if you extrapolate from that, what you might predict is that some aspects of normal aging may relate to lamins, but not all of them. So I think we will learn things about aging from studying progeria. I'd also point out that, that companies that are right in our backyard, like BioMarin and Ultragenyx, have figured out ways to develop drugs for these rare diseases and make money off of it. And so there are strategies to go forward to develop therapies. And, I was of the opinion when I started doing these studies that I was just interested in progeria to understand normal aging. But then I started meeting these kids with the disease, and as soon as you meet them, you're like, I got to do something about this.
Brian Kennedy, President and Chief Executive Officer of the Buck Institute for Research on Aging. Brian, thanks so much for your time today.
Anytime. Thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.